Welcome to Simplified UPSC by PPP, where we listen, learn, and grow together. Detailed lecture notes in English and Hindi, plus practice questions for prelims and mains, only on our Telegram channel. Link in description. Like and subscribe for more.
Welcome back to the deep dive. If you're here to cut through all the information overload and, get that deep synthesized knowledge fast, especially when you're prepping for exams like the UPSC, then you are in exactly the right place.
Today, we are going deep. I mean, really deep on probably the most foundational chapter in all of biology, NCERT Class 11, Chapter 8, The Cell, the Unit of Life.
And this isn't just about memorizing parts of a diagram.
Not at all. It's about understanding the core principles that connect, well, everything, from ecology to human disease. We're going to show you the critical links that often get missed in a first read-through.
Before we pull out the microscope, though, a huge heartfelt thank you. Seriously, to all our existing subscribers and the paid members of the Simplified UPSC by PPP YouTube channel, your support is what makes this kind of detailed analysis possible.
It really is.
And if you're listening and you find this content genuinely useful for your prep, and if you're in a position to, please consider subscribing, hitting that like button, dropping a comment, and maybe even joining the paid membership. We truly appreciate it.
We do. Thank you.
Okay, let's get into it. The UPSC asks you about, well, everything. 1 minute, it's optical illusions. The next, it's geology.
Right.
Then you get a biology question, maybe a practice one, asking about biofertilizers. And it lists, say, agaricus, nostoc, and spirogyra.
And that seems so specific, so random, but it brings us right back to the cell. The answer is Nostoc, which is a cyanobacterium.
Okay.
And it perfectly shows how a simple cellular design can lead to this huge, complex ecosystem function. Nostoc is a nitrogen fixer, but the enzyme it uses is inhibited by oxygen.
Which is a problem since it does photosynthesis and produces oxygen.
Exactly. A huge problem. So how does it solve it? It uses cellular specialization. It develops these special thick-walled cells called heterocysts.
Wow.
These cells don't do photosynthesis. Their whole job is to create an oxygen-free zone just to fix nitrogen. The entire nitrogen cycle, which, underpins agriculture, it all hinges on the design of that one tiny cell.
That's the whole point right there. Complexity comes from simplicity. And speaking of simple definitions, our sources say the defining feature of life, above growth, metabolism, all of that,
is cellular organization.
Cellular organization. If it's alive, it has cells, period.
Precisely. And the simplest form of that organization is the prokaryotic cell. Think bacteria, blue-green algae, mycoplasma.
And they're defined by what they don't have.
That's the easiest way to think about it. No well-defined nucleus. And, critically, none of those membrane-bound organelles. No ER, no Golgi, no mitochondria.
So if there's no nuclear membrane, where's all the genetic info? Just floating around.
Not exactly floating, but it's concentrated in a region we call the nucleoid. The key term here is that the genetic material is naked. It's not protected by an envelope.
Okay.
And many bacteria also have these extra small circular DNA molecules on the side. They're called plasmids.
And these are important.
Hugely important. They often carry the genes for things like antibiotic resistance. This is fundamental for understanding modern medicine and genetic engineering.
So the cell's survival really depends on its outer defenses. The cell envelope, you said it's a 3-layered structure.
A formidable one. Tightly bound and chemically complex. The outermost layer is the Glycocalyx.
And its texture matters.
It does. If it's a loose, sticky sheath, we call it a slime layer. But if it's thick and tough, it's a capsule. That capsule is what helps some nasty bacteria evade our immune systems. Another direct link to disease.
Got it. Layer 2 is the cell wall. That's the skeleton.
A structural support. It gives the cell its shape and, crucially, stops it from bursting or collapsing under pressure.
And then layer 3, right underneath, is the plasma membrane, the gatekeeper.
The selective barrier, yes. And it's the subtle differences in this whole envelope that let us classify bacteria with the gram stain.
Gram positive or gram negative?
Exactly. Whether it takes the stain or not. And this isn't just for textbooks. In a hospital, that result can literally decide which antibiotic a doctor uses. That's applied cell biology right there.
So a quick question. Prokaryotes don't have mitochondria for respiration.
Yeah.
How do they generate energy on a large scale without all that internal membrane surface area?
They improvise brilliantly. They have mesosomes.
Mesosomes.
These are just extensions of the plasma membrane that fold inwards into the cell. They help with DNA replication and making the cell wall, but their biggest job is respiration. They increase the surface area for enzymes, basically acting as a stand-in for the mitochondrion.
A cellular workaround, I like that. What about movement? I hear about Flagella, Pili, and Fimbriae. Are they all for swimming around?
A classic point of confusion. The only one for motility, for movement, is the bacterial flagellum. It's got 3 parts: filament, hook, basal body, and its only job is to move the cell.
Okay, so what about the other two? Pili and Fimbriae.
The sources are very clear on this. They do not play a role in motility. They are for attachment. Fimbriae help bacteria stick to rocks or, tissues in your body. And Pili are involved in bacterial conjugation. So one moves, the others stick.
So if the prokaryote is this model of elegant efficiency through simplicity.
Right.
Then we get to the eukaryotic cell. Quotists, plants, animals, fungi. And this is where things get really interesting because it's all about complexity, all about compartmentalization.
That compartmentalization is everything. It lets all these different complex processes happen at the same time without interfering with each other. But not everything inside is wrapped in a membrane. Let's talk about ribosomes.
The protein factories.
Protein factories. They are non-membrane bound, and you find them in both prokaryotes and eukaryotes.
But they're different sizes. Prokaryotic ones are 70S and ours are 80S. Why does that number, that Svedberg unit, actually matter to a student?
Because it's fundamental to medicine. Many of our best antibiotics are designed specifically to target and shut down the smaller 70S ribosomes in bacteria.
So they kill the bacteria.
Without harming the 80S ribosomes in our own cells. It's a precision strike based entirely on a tiny difference in subcellular architecture.
Okay, let's move to the real power players. The powerhouse and the food factory. Mitochondria and plastids, like the green chloroplasts.
These two are all about energy. Chloroplasts trap light energy. The light reactions happen on these flat, stacked membranes called thylakoids.
Stacked in the grana.
Right. And the dark reactions, the Calvin pathway, that happens out in the fluidy part, the stroma.
So here's where it gets really cool, the synthesis part. What's the high-level insight about mitochondria and chloroplasts?
It's their shared history. They are unique. They divide by fission, just like bacteria do.
And the big one.
And the big one. They have their own small circular DNA and their own smaller 70S ribosomes.
Just like a prokaryote.
Exactly. This is the core evidence for the endosymbiotic theory. The idea that these organelles were once free-living prokaryotes that got engulfed by a bigger cell. They are basically tiny bacteria living inside our cells, which is why they have their own DNA and their own simple ribosomes.
Amazing. Okay, shifting from energy to architecture. The eukaryotic cell is complex in shape, but it doesn't have that rigid cell wall like a bacterium.
Yeah.
How does it hold its shape and move things around inside?
It uses its cytoskeleton. You should think of it as the cell's internal scaffolding, its highway system, and its muscles all rolled into one.
So it's a network of proteins.
An elaborate, dynamic network of filamentous proteins, specifically microtubules, microfilaments, and intermediate filaments. It gives mechanical support, maintains the shape, and it's responsible for motility inside the cell, literally dragging organelles from place to place.
And for moving the whole cell, we have cilia and flagella here too.
We do, but they're much more complex than the bacterial version. They grow out of structures called basal bodies. Cilia are short, numerous, and they move like oars, often to move fluid over the cell. Think of the ones in your lungs clearing mucus.
Right.
Flagella are longer, and they propel the whole cell forward. Totally different internal structure based on microtubules.
Okay, so now we have the structure. Let's connect it to the function, the chemistry that drives it all. Metabolism.
And why this matters for the UPSC is how we classify the molecules involved. First, you have primary metabolites.
The basics. Amino acids, sugars.
Mm-hmm.
The stuff you find in all animal tissues.
Exactly. But then you have the secondary metabolites. Things like alkaloids, essential oils, rubber.
The ones you find in plants, fungi, microbes.
Right. And their economic and medical importance creates a direct link between cell biology and bigger topics like forestry, industry, and geography.
When we break down a cell chemically, we get different fractions. There's the acid-soluble part with the small molecules.
The micromolecules.
And the acid-insoluble part with the big ones, the biomacromolecules. Proteins, nucleic acids, polysaccharides.
There is a critical exception here that is a favorite for exam questions. Lipids.
Lipids. They're small molecules, they're micromolecules, but they end up in the macromolecule fraction.
Yeah.
Why is that?
It's because of where they live. They're part of cell membranes. So when you grind up the tissue, the membranes shatter into tiny vesicles that aren't soluble in acid. The lipids are trapped in those fragments, so they get pulled down into the insoluble biomacromolecule fraction, even though they aren't big molecules themselves.
That's a great distinction. And driving all of this chemistry, enzymes.
The catalysts, fundamentally, they're proteins. And we need to stress this. There is no such thing as an uncatalyzed metabolic reaction in a living system. It just doesn't happen.
And the way they work is the catalytic cycle.
Simple and perfect. The substrate, or S, binds to a very specific spot on the enzyme, the active site, or E. They form a temporary ES complex. Then, boom, the enzyme releases the product, P, and the enzyme is unchanged, ready to go again.
And these enzymes create pathways. Anabolic pathways build things up like proteins, and they consume energy.
While catabolic pathways break things down, the classic example is glycolysis, the 10-step breakdown of glucose. That releases energy.
And that released energy is captured
and stored in the universal energy currency of all life, adenosine triphosphate, ATP.
So if enzymes drive everything, what happens if the cell's environment changes?
They are incredibly sensitive. They only work in a very narrow range of temperature and pH. If you go too far outside that range, the enzyme's 3D structure starts to unravel. It's a process called denaturation.
And that means the active site changes shape.
Exactly. The substrate can't bind anymore, and the reaction just stops.
So when a person has a very high fever, the danger isn't just feeling bad, it's that the high temperature could literally start denaturing critical enzymes, shutting down metabolism.
That's the life-threatening risk. It's a failure of the basic cellular machinery, a powerful link from basic chemistry to clinical medicine.
Wow. So this has been a journey. We've gone from the simple, efficient prokaryote with its envelope all the way to the specialized eukaryotic cell with its captured organelles. And finally, to the chemical engine room of metabolism, all powered by these incredibly sensitive enzymes. You should now have a really synthesized view of how the cell works and why it matters everywhere.
And to leave you with one final thought, something to chew on that builds on all this, think about the alternation of generation in the plant kingdom.
The switch between the haploid and diploid phases.
That whole complex reproductive cycle, from simple algae all the way up to flowering plants. What is it, really? It's nothing more than a perfectly timed, meticulously orchestrated sequence of cell divisions and differentiations, mitosis and meiosis. The entire drama of plant evolution is a story told by individual cells using the exact machinery we just discussed.
The cell really is the blueprint for everything.
It is.
Well, if you found this deep dive essential for your UPSC prep or just plain useful, please remember to support the Simplified UPSC by PPP YouTube channel. Subscribe, hit the like button, share your comments, and if you can, please consider joining the paid membership.
And a huge thanks again to all our existing members. You make this possible.
Thanks for watching. Follow us on Telegram and Instagram. Don't forget to like, share, and subscribe.